with some good information. Many of us, if any people in the room who were around in the mid-'80s, early to mid-1980s, when HIV testing first started, although we called it HCLV3 testing at the time, know that we had an antibody negative window of 6 to 12 weeks or so, and as new generations of HIV testing came on, that's now down the testing negative window. I won't say antibody, but testing negative window now, somewhere between 2 and 3 weeks, as you'll hear from our speakers. So I'll let them do all of the details, but again, welcome. It's good to have you all here, and I'm honored to be here with the speakers that we have. And at this point, I'd like to introduce the first speaker, Dr. Mark Pandori, and I'm really impressed that when in meeting Mark the first time, because some of you, particularly if you're from Atlanta, may know that Mark used to play for the Braves, and that he was the MVP of Major League Baseball in 1998. He had a batting average of .343, he had 24 home runs, and he missed the Triple Crown by 3 RBIs. And if some scorer hadn't called one hit an error, he probably would have had those 3 RBIs. So sorry about that, Mark. But really, though, Dr. Pandori, currently, though, he's now gotten into a better field. He's Director of Laboratory Services for the Alameda County Department of Public Health in Oakland, California. He's the CLIA Director of the San Francisco Department of Public Health Laboratory and an Associate Clinical Professor of Laboratory Medicine at the University of California. Dr. Pandori currently serves on the Infectious Disease Committee of the Association of Public Health Laboratories. So it's an honor for me to present Dr. Mark Pandori for our first talk.
Thank you, Tom. Can everybody hear me all right? All right. So I'm making a lot more money now working in lab diagnostics instead of Major League Baseball. Thank you very much. Excited to be here. Certainly the crowd's a lot smarter. So I hope that those of you that visited, of course Bio-Rad employees know, but those of you that are visiting the meeting, the AACC picked up a Bio-Rad pen at the Bio-Rad exhibit, unless you're from Massachusetts, Vermont, and what's the other one, Rhode Island? But I consider this a symbol of Bio-Rad's innovation because, as you may know, if the ballpoint is out, you can't put this in your pocket. It's got a safety on it, and it might be one of the only ballpoint pens like that. So there you go. I consider it a symbol of Bio-Rad's skills in designing laboratory or equipment. So we've been dealing with HIV as a medically recognizable entity now for over 30 years. And while we have no vaccine and we have no cure, testing really remains our only weapon against HIV right now. I mean, we have good treatments, but in terms of preventing infection and dealing with infection, getting people into medical care, it's really testing is our best weapon. And when we talk about HIV testing, if you look at my background, I always have to see HIV testing through 2 separate lenses. I look at it through a medical lens, which is we test an individual to find out whether they are unwell so that we can provide them with medication to make them as well as we possibly can, and that's medical care. I also look at HIV testing, because I'm from the public health realm in particular, through a public health lens, and that is that we test people because what we're really interested in public health is the elimination of transmission networks. So we're actually looking at testing as a way of preventing spread of a disease across the population. As laboratory diagnostic scientists, however, I think this is an interesting topic all by itself without either of those other 2 reasons, and that's because with HIV, we have an ever-evolving field of diagnosis. We have constant evolution of technology, and we probably have more screening tests for HIV than we have for any other infectious disease, at least in the FDA-approved realm. And not only that, we can't use these tests willy-nilly, as you're going to hear later on from our other speakers. We test for HIV according to recommendations and algorithms. So there's a lot of complexity to this topic, and it's one of my favorite topics in the field of laboratory diagnosis, and that'll probably come across a little bit. And so I welcome you again, and I'm going to launch into my part of it, which has to do with screening for HIV infection. So in terms of screening for HIV infection from an FDA-approved point of view in the United States, we really have 2 options. We have laboratory-based tests. Often these are enzyme immunoassays, but we might just be safer if we call them immunoassays, because some of the screening tests might in fact be chemiluminescent immunoassays instead of enzyme-mediated. We also have point-of-care rapid tests. And I have pictured examples that are very familiar to all the diagnostic scientists here. You can see kind of a typical EIA and what your typical point-of-care rapid test. And an OraQuick is probably maybe the most common representative of that family of tests because it happens to be the one that you can buy over the counter. I'll launch right into the evolution of screening tests on the lab side of things. On the lab side, again, enzyme immunoassays have really been the workhorses for screening for HIV infection since around 1985. And the first generation of these tests that we had, what we call, and we use the terms generations to refer to screening tests in the HIV diagnostic field. The first generation of these tests, they were created by lysing whole virus particles, welding them to the bottom of an ELISA well of a 96 or larger plate, and then using that as capture antigen for antibodies. And so these tests didn't really reliably detect antibody to anything but the strains of the viruses that were used to make those tests. And what that translates to here is they didn't really reliably detect antibody to all HIV-1 strains or antibody to HIV-2 very reliably. These first generation enzyme immunoassays only detected IgG, and the window periods of these tests, as was alluded to in the introduction, were probably around 6 weeks. The idea of window period, as was alluded to a little bit earlier, is I'm going to pound on that in my part, and I think it's probably going to come up again. It's becoming, in some ways, the most important aspect of HIV testing for a number of reasons. And I'll talk more about it in a few slides, but a window period is essentially how long somebody would have to be infected from the time they're infected. How long would they have to be infected before the test would have a very good chance of detecting that infection or evidence of it? And so these tests had pretty long window periods of about 6 weeks on the average. They're FDA approved, at least one of them is FDA approved for use with oral fluid testing and dried blood spots as well. So these tests actually, not only could one test blood, plasma or serum, but blood spots and oral fluid as well. We evolved to second generation tests, which were essentially the same, but your capture antigens were generated by recombinant DNA technology, or they were recombinant proteins. And so this allowed an increase in specificity and a concomitant increase in sensitivity. Again, they only detected IgG, but these increases in sensitivity and specificity allowed us to have a test that had a window period of about 5 weeks. We really made a large leap in the 90s, late 90s, early 2000s, when the third generation tests came about. And these tests use a different mechanism of antibody detection. And with third generation enzyme immunoassays or immunoassays, we can detect IgM and IgG. They detect reliably antibodies to group O HIV and not just group M, and antibodies to HIV-2, although they do not differentiate between HIV-1 and HIV-2 antibodies. The window periods for third generation tests were a pretty big leap. They're on the order of 3 weeks or 22 days. So that's a much shorter amount of time that a human being would have to be infected before a test could say so. And again, that's going to just keep coming in this talk. We went in June of 2010, we finally got FDA approval on a couple of fourth generation tests. These screening tests are a significant advancement in that they simultaneously detect antibody to virus and antibody. They detect antibody and virus simultaneously. The two that are currently FDA approved that we refer to as fourth generation tests don't differentiate that. So when the positive result occurs, they're telling you that it's positive, but it could be antibody, it could be virus. They detect the antigen of the virus. They're not RNA tests, and we'll talk a little bit more about that in a bit. The advantages of detecting antigen and antibody are that you can detect recent and acute infection that lowers the window period. We'll get into that, as I said, in a few slides. They can have faster turnaround times. At least one of them is a random access testing device. They're cheaper than an RNA test, and they're FDA approved. RNA tests, which I'll get into in a little bit, are very sensitive for recent infection, but they're not FDA approved for that purpose. Let's go into the rapid testing options. So we went through the generations of the evolution of laboratory tests. Now we'll look at what has happened on the other side. In the public health realm, the vast majority of testing actually occurs using point-of-care testing. When point-of-care testing came along, and I was at the public health lab in San Francisco, I'd say 90% of our business went out because so much of testing got pushed out into the field and into medical offices and into outreach. When we talk about point-of-care tests or rapid HIV tests, we're talking about tests that are waived in terms of their complexity rating to CLIA. That is that you don't need a licensed individual to run these tests. So they're routinely done outside of a laboratory. They can be done in doctor's offices or in outreach clinics, or sometimes there's mobile testing in public health. And because a licensed individual is not required to run that test, you can actually broaden the amount of people that get tested. You can test more people more quickly instead of having to go through the laboratory process. Some clinical labs actually do use rapid tests, and actually for a while San Francisco General Hospital even used a third generation rapid test that I'll get into, and that allows you to get rapid results, so you can actually do a stat HIV test a little easier instead of waiting to batch. You know, a lot of us that work in labs, if you have an automated piece of instrumentation for running HIV, you would wait to batch your testing, but with these you wouldn't necessarily have to do that. You can run them much more quickly and more flexibly. The OraQuick can use blood or oral fluid. The sensitivity is comparable to that of a first generation EIA. It takes about 20 minutes to get a result. You have the ClearView Stat Pack, which allows a similar sensitivity to the OraQuick, but it only uses blood. You can use plasma or serum on a test like this in the laboratory. It becomes moderately complex when you do that. So a licensure would be required. This one is notable. This is an FDA-approved test called the INSTI, which is a rapid test which gives you results in just a couple of minutes. So in public health, we worry about people returning to get their test results sometimes, but if you promise they're going to get their result in just a couple of minutes, you can often get people their results very quickly and very reliably. The Unigold is a rapid test, which is interesting only in that we call it, let's say, so to speak, a third generation test, in that and that's probably not the right nomenclature, but it detects IgM and IgG. So you get slightly better window periods with this test compared to other rapid tests as screening tests. There's the allele-determined test, which you might call a fourth generation screening test, which is at point of care. This detects antigen and antibody and differentiates between the two, although its sensitivity for antigen is probably not incredibly high, at least can detect perhaps some acute infections. The multi-spot is actually FDA approved as a screening test as well. This is the only FDA approved test in addition to the Geenius that can detect antibody to HIV and 1 and 2, but differentiate between the two as well. So when we talk about HIV screening tests, though, we really have to think a lot about window periods. And I wanted to show you this slide, which shows the course of HIV infection from the time of infection onward. And what you see, the very first thing that happens when exposure and infection occurs is that viral load goes up very quickly. And first thing, and viral load skyrockets, and you'll get very, very high viral loads that you may not see ever again in the course of infection, extremely high, into the millions of copies per mil. After a few weeks, as was implied by the data I showed you earlier on some of these screening tests, you'll see antibody development at around week 3. And so you'll see tests that are sensitive for IgM becoming positive at about week 3. But you can see there is a 3-week period there where a virus is present and replicating unchecked, and there's no antibody to detect. And I drew a line to sort of show you quite kind of around that area. So again, it brings us back to window periods. And if we wanted to look at all these tests on a timeline, your rapid IgG sensitive point of care tests are out here at around day 40-ish. Your lab-based tests and maybe your allele rapid test are maybe around day 22. Your fourth gen tests are hovering around day 16. And your confirmatory tests are way out here. And I'm going to let Barry talk to you about confirmatory tests. But the fact that that early on, the sensitivity of those tests, and even now, I think with multi-spot, the sensitivity of those tests has window periods out there, and some of your screening tests are way here. There's a gap there that you have to worry about and be mindful of. But I'm going to let my cohorts talk to you about that. So we really do want to talk about recent and acute HIV infection and the laboratory diagnostics of it. When we talk about acute, I think the thing we all agree upon in this definition is that we're talking about recent HIV infection. For diagnostics, a refined definition might be pre-seroconversion, and this would be maybe around 3 weeks to, in some patients, as much as 6 weeks before antibody is developed. And that might have something to do with what test you're screening with as well, whether it's 3 or 6 weeks. And again, so I'll show you this slide again, but I'll add the fact that that viral load early on that we're talking about correlates very well with viral load in semen, and so there's consequences of that. Also, during that recent period, that first 3 to 6 weeks, a patient may present medically with a syndrome that's mononucleosis-like or influenza-like, and so And it is important for us, I think, to have tools available to us to detect recent and acute infection. Here in a study done in, I think it was Uganda, where we looked at serodiscordant couples where one person's HIV positive and one person is HIV negative, and looking at how many copulatory acts it might take in order to transmit HIV. And what you can see is that during the acute phase, transmission of HIV occurred much more readily, orders of magnitude more probabilistically than it might occur for people that are chronically infected. So after viral loads come down and a person's immune system is kicked in and the viral loads come down, the probability of transmission decreases incredibly. So from someone like me who looks at testing through a public health lens, now you may start to appreciate why window periods are so critical to us, because we want to catch people, or at least have a chance to, when they're most infectious. So in order to do that, I think from what you've seen, then you would say, well, gee, what about the direct detection of virus? And I will tell you that the tools at your disposal to do that are RNA detection and fourth generation testing. And we're going to talk about something else after that. Viral load testing is available. We're talking about RNA testing. The tests that we think about when we hear about viral load, we detect and count HIV RNA. We're probably thinking about RT-PCR, quantitative RT-PCR. They quantify virus in the blood. They're typically used for patient management. They're only FDA approved for that purpose. Looking at the efficacy of a person's treatment, whether they're taking their drugs, whether the drugs are working, looking at viral load, perhaps it's done perhaps every 2 months with an HIV-infected individual. They are FDA approved only for patient management. They're not approved as diagnostic tests. One RNA test does exist. I don't know if anyone else is going to talk about it, but it is FDA approved as a confirmatory or a supplemental test, but not as a screening test. It's not widely utilized. Your real-time PCR sensitivities are on the order of about 40 copies per milliliter. So that would be able to detect infection pretty early, as you can imagine. In San Francisco, at the Department of Public Health since 2003, 2004, thereabouts, we started doing screening of people using RNA. So we knew we had a high-risk population and that using antibody testing and the concept of window period, that if people came and got tested in the middle of that window period, they would test false negative on an antibody test. So we wanted to be sure that we had a chance of catching high-risk individuals. And so we did that by pooling every 10 antibody negative specimens into one specimen and testing it for RNA. And then if that specimen is positive, going back to the constituents and finding out which of the constituents was positive for RNA. And to date, we've captured over 220 individuals who were caught on an RNA test and missed on the initial antibody screen. For some of those that were screened by something like a point-of-care rapid test, we found that about 44% of those people actually would have been caught if a high-quality test would have been run in a laboratory with a window period equivalent to that of a third-generation test. But about 55% of them would have only been caught by RNA. So those are people that are very recently infected and very infectious because of where they are in that window period. You have antigen antibody diagnostic tests like fourth-gen test, which I showed you earlier. P24 detection is what's at the core of a virus particle. It's also at the core of the fourth-gen's methodology. The P24 protein is a protein which exists at about 1,200 copies per virion in the middle of a particle. A fourth-gen test can detect that protein in addition to IgM and IgG. In a study that we did in San Francisco looking at acute specimens, so these would be specimens that are absent, they're totally negative on all antibody tests but positive on RNA tests, we see that the sensitivity of a fourth-gen, the architect for virus was somewhere on the order of about 30,000 RNA copy equivalents. So it's, if you compare that to an RNA test, which has a sensitivity of 40, you can see quite a difference. But of course, during that early phase, RNA is actually rising relatively quickly during the acute phase, and you do tend to see higher viral loads. So another paper that looked at the architect, which is a fourth-generation test, said that sensitivity was probably on the order of around 50 or 60,000, which matched our data fairly reasonably. Our data was generated with clinical specimens, and this data was generated in virus that was grown in the laboratory. But what about the fifth gen? Now, we had news that there was our FDA approval on a test on the Bio-Rad Bioplex platform, coincidentally. And on that platform, you can differentiate when you detect, when you get something positive, it will tell you whether it's positive for antibody, for HIV 1 or 2, or it'll tell you if it's positive for antigen, which is something that fourth generation tests don't do. And it looks like from some preliminary data that the sensitivity for antigen is a bit higher than it is for the currently FDA approved fourth generation tests. So if you were to translate that into RNA copies, as I just told you, if a fourth generation test has a sensitivity of about 50,000 RNA copies per mil, the data is indicating that perhaps this test might have a sensitivity around 14,000. Does that translate into a window period differential? And the question is probably yes. If you look at the kinetics of viral replication in an infected individual and you look at what the doubling time of RNA is in a newly infected individual, that might translate to a day of window period, but we're not sure, and we'll have to really do the studies to find out. So if you had to guess, you might say that the Bioplex test would have an even better window period. It would be somewhere between that of an RNA test and a fourth generation IA. And now I'm going to take you on to supplemental testing, and then you're going to hear about testing algorithms. And I will hand it off to my friend and colleague, Tom, and you'll do. Thank you.
Thank you very much, Mark. That was great and very informative. Okay, thank you. Move on to the next speaker, and this is Barry Bennett, who's coming to us from Jacksonville. And Barry's been a virologist for the Florida Bureau of Public Health Lab since 1977. He formed the retrovirology unit in 1985, and since the mid-90s, the department has been proactive in developing and providing a comprehensive HIV clinical management testing program, in addition to advanced HIV 1-2 antigen antibody diagnostic testing services. The Florida Bureau of Public Health Laboratories is proud of its commitment over the past 30 years in providing state-of-the-art HIV diagnostic and disease management services to their Florida clients. I don't know that Barry actually was on the baseball team on the Florida Marlins when Mark was playing for the Braves. But Barry and his wife are very active in the Jacksonville Zoo and Gardens and volunteering and working in the community services from that standpoint. And as an animal lover and a zoo lover, I'm very, very pleased to hear that. So it's a great pleasure. I'd like to introduce Barry Bennett. Okay.
Thank you, Tom. And to add to Mark's, my extent of baseball is probably related to just softball. And when I get a hit, I get to first base, and then I raise my hand. I want a pinch runner from their own base because I'm just their pitcher. So, but yeah, it's a pleasure to be here tonight, and I want to thank the Bio-Rad group for the invite and for sponsoring this talk. A little bit about, as Tom said, I'm with the Florida Bureau of Public Health Laboratories. Been there my whole career. I'll be there for 5 more months. And I went to my lab director today, and I'll be retiring after 38 years. And I told her, I said, well, what do you want me to do with my email account? She says, well, what's the rush? You know, just keep it for another 6 or so months. You may need it. So Tim's probably going to still be getting emails from me come 2016, but you never know. What I wanted to just to add on to what Mark presented and talk a little bit about the differentiation test and some recent data we've had with the most recent differentiation test in the U.S., the Geenius HIV 1-2. Here's 3 documents that I really recommend those of us in infectious disease testing and particularly HIV that you should have these on your desk or readily handy. And of course, the one on the far left is the guidelines from last year from CDC, and then APHL in cooperation with CDC, and actually on our very last one with ASM, have been collaborating into producing some other documents to help laboratories and clinicians adopt the new algorithm and what it's all about. I know I was talking to John earlier today, and we were talking about, some people say, well, where did this algorithm come from? I mean, I haven't heard anything to it just happened. And was it just a few of us went together and just sketched it out, and there it was. But no, this was 8 years in the making, right, Michele? 8 to 9 years in the making. So a lot of supporting evidence has gone into it, and we're just going to scratch the surface of it tonight. But the document in the middle is a document specifically for reporting language, both to the clinician and to surveillance for those of us that have to report, to a surveillance type and setting in our states. And the most recent was the one on the far right, released just this past June, on really we still saw a need that people were, it was hard to pry them away from their dry blood spots. I mean, not their dry blood spots, but we could talk about that later too, but their Western blot. And we've put this document out to further help those that want to transition from Western blot into one of the new differentiation tests. And I'll show this again in case you didn't get all those links. You'll have another shot at it. So my objective is to review the supporting evidence for the use of the differentiation test and the new algorithm, and also to discuss, like I said, some recent data with the retrospective study we performed on the Geenius 1-2 supplemental assay. So this is the area of the new algorithm. In case you're not familiar with the new algorithm, this is it. But and I'm going to focus strictly on that second test of now we have a 3-test algorithm, and my talk will be focused on that. And if you have any other questions about the algorithm, I'm sure any of us up here would be glad to field that. So as of today, I guess, currently we have 2 FDA-approved differentiation immunoassays that fill that role in the new diagnostic algorithm. We have the one on the left is the Geenius HIV 1-2 supplemental differentiation assay. It actually has a supplemental claim. It was approved in October 2014. Both of these assays are moderately complex, CLIA status. The Geenius test on the left is more of a DPP, dual path platform format. As you can see, it has a 2-dimensional barcode for the cassette, so you have positive identification on the cassette, and if you add right below it, you may see an area sort of raised where you could put a barcode of your patient or your client, and so you'll have basically a double positive identification for this. And you also see that there's 7 markers or 7 places where you can see the reaction take place in the window. We have 2 peptides for HIV, 2, GP36 and 140, and then we have 4 HIV-1 antigenic proteins. All of these are synthetic peptides, except for the 160 and the 24 are recombinant. This is read by an automatic reader, and before you even ask, there's no way of putting the cassette in the wrong way or upside down. We tried it. It won't go in. So it's foolproof there. It'll read. It has its proprietary software where after the reading, it'll go over for a report for you to review and confirm, give you a little picture of the reading of the cassette, and produce your report. On the right, it's a multi-spot HIV 1-2 differentiation test, and as Mark said, it was FDA approved for screening test, and then it went back and received an additional approval in March 2013 with an intended use statement specifically designed to help us to use it in the new algorithm as a differentiation test for the algorithm. And also with that, we also saw that an indeterminate status for a result was added to that. We're basically dealing, for those of you who are not familiar with it, but I'm sure a lot of you are, you have both recombinant and a peptide for GP41 and a peptide for 36. Speaking of the still on the multi-spot, with that revision in the intended use came the indeterminate status. And here's where I want to draw some distinction between the Geenius and the multi-spot. You'll see with the multi-spot, the terminology is HIV positive undifferentiated. When you have both, as you can see it pictured, representation of reactivity with HIV-1 and HIV-2. And there is a dilution protocol that is available to try to resolve that with the multi-spot, but it is not available on the Geenius. And also, you'll see the HIV-1 indeterminate status, and that is when you're really only seeing a recombinant or a peptide for HIV-1 reactivity, so then you should follow with further testing. In this case, would go to a qualitative NAT test per the algorithm. So a couple of questions have come up. We've actually done, through the APHL, national webinars on frequently asked questions, and again, to help promote the algorithm in the country. And one of the questions is, why do we want to replace Western blot with the differentiation test? And we have several people in the room here, Michele, and I think I've seen Sylvania's here, some of the work they've done at CDC, and also covered again in the guidelines that it detects, the differentiation test detects HIV-1 antibodies earlier than Western blot. And primarily on this example of the 183 specimens from 15 seroconverters for HIV-1 and 2 differentiation IA, this happens to have been the multi-spot, they became reactive 7 days before the HIV-1 Western blot. And of course, this also leads to fewer specimens needing to be further tested by NAT because of the concordance between the first and second test. The guidelines also gives as many as 9 studies to support the high specificity of these tests compared to blot. And I think Mark even had a recent article in Medical Lab Management talking about the reagent costs are sometimes as much as less than half for these differentiation tests compared to Western blot. Then we have a faster turnaround time. You're looking at basically on-demand, not point of care, but rapid differentiation testing as compared to batching for a Western blot. And we have more accurate diagnosis now for HIV-2 infections. And to expand on that, there's been at least 2 large studies, one out of New York and another one published in the MMWR in 2011, that anywhere from 60 to 93% of the HIV-2 individuals were misclassified as HIV-1 Western blots. So unfortunately, these HIV-2 cases, we're not even diagnosing them until we get a negative viral load. So this is after they've sort of moved out of the diagnostic realm of their life into clinical management. They found out they're really not infected with one, and all of a sudden now you have to back them back up and do HIV-2 confirmation before we get them into care. So this is a waste of resources, a waste of time, and a benefit to the patient. A little bit about turnaround time. I thought I'd share some of our data on this. We transitioned to the new algorithm in April 2012. I knew it was coming. We had time. We had opportunities to do it. So we went ahead and did a double shift. We went from a 3rd generation to 4th generation, and we went from Western blot to multi-spot. So we wanted to look at our turnaround time. So we looked at the 3 months prior to April and the 3 months after. And from January to March, less than about 22% of our positive cases were reported in less than 2 days. After the transition, from May to July, 96% of our HIV-1 positive cases were reported in less than 2 days. So a dramatic turnaround and enhancement in our turnaround time and release some positive results. So why not go directly to NAT after your primary screening test? That's been a question that's come up. And cost is one of the first ones you would think of. Most of these specimens we deal with are resolved with less expensive antibody testing. And the turnaround time for confirmed tests is, until we get to the days of rapid NATs, this is still probably a less than 90 minutes to screen and confirm with the current algorithm. It fails to diagnose acute infections. You need that antibody step in there, testing step, to help you confirm those acute cases at stage 0 for surveillance people. Or in our HIV-2 cases, we need that differentiation in there. And that is undetectable in 3 to 5% of the Western blot positive specimens. We know this is really consists of elite controllers and also viral suppression from long-term antiretroviral use individuals. So a little bit now about the study that we recently wrapped up. We looked at comparing the Geenius HIV 1/2 supplemental test to our multi-spot, and we looked at 350 well-characterized specimens. 250 of those were established HIV-1 positive antibodies. So we consider these as fourth-generation combo testing. Oh, by the way, do you know the difference between combo and combi? We learned this at the last diagnostic conference from our FDA colleague. Anybody know the difference? Combo versus combi. Combo is an antigen antibody test. Combi is an HIV 1/2 antibody test. For what it's worth. But anyway, so we tested with our combo test. It was repeatedly reactive. I'm right, Michele? Okay, I'm right. Okay. And then in our differentiation test, the multi-spot was reactive, HIV-2 was non-reactive, in case you're interested in that. And we stop right there. So those are our established infections. And that was the largest pool of our group. We had 60 false positive combos. And the key here is, yes, the combo was repeatedly reactive. The multi-spot was either non-reactive or indeterminate, and I'll have the numbers for that in a second. But the key is the HIV-1 NAT was non-reactive. And to the best of our ability, we made sure that there was no antiretroviral use individuals included in this group. And then we have 20 what we call acute stage infections. In our public health lab, we have a very, very narrow definition of what we call an acute stage. In fact, if you look at this number 3 and 4, most laboratories would not separate those out. They would consider both of those acute infections. But we like to look at those that are clearly combo positive, multi-spot non-reactive, or differentiation non-reactive, and then NAT positive. That's our acutes. And then if you see beginnings of antibody production, and hopefully that's represented by the differentiation test of being HIV-1 indeterminate, then we call that an early infection. Definitely worth follow-up, get these people into care right away, partner notification, but not quite to the highest degree as we do acutes. And then the last group that we looked at were some known HIV-2 antibody positive specimens that Bio-Rad provided us. So this is the protocol. Most of you, I feel like I'm preaching to the choir to most of you on this one, that you've got a lot of experience to it, but if you're not, it's 5 milliliters of serum or plasma, or you can use whole blood. All of our specimens were serum and plasma. And then you add the buffer, 2 drops of the buffer, wait 5 to 7 minutes, waiting for all the color to migrate across the screen. And then you add 5 drops of the buffer at the other addition, wait 20 minutes, and after that you have up to 30 minutes to place the device into the reader and to record the results. I'm not going to go too much into the details of this. I understand it is at on the floor of the expo, and there's plenty of individuals that will sort of go over that with you. This probably is not real visible, but this is right out of the package insert of the interpretive criteria. We have both the global, let me find my pointer here, our global interpretations on this side, and then the individual HIV-1 and HIV-2 interpretations. I'll have another slide that shows these a little bit more in detail. But I want to draw your attention that we have 2 that have a little bit more to think about, the HIV positivity with HIV-1 cross-reactivity. So you are seeing both HIV-1 and 2 reactions, but the HIV-1 is only the 160 or the 41 proteins, but not both. If you had both proteins, then it would be considered, along with the HIV-2 reactivity, as an untypable. So we'll go over that again in detail. So here's the results broken up by HIV-1 versus HIV-2. You can see the different categories that we would have as a possible result. Again, the global interpretation is on the left, and the individual interpretation is in bracketing. So you have one HIV-1 positive indeterminates, and of course HIV-1 negative, and several scenarios of each of those. So let's look at our comparative data of those 5 different categories. For our established infections, we had 250, and we had 100% agreement on the initial test to the multi-spot results. Now we were also, we had 23 samples that had some degree, they were either 4 were untypable, and we had 19 that also had HIV-2 indeterminate results. And as the package insert calls for to retest in a new cassette, 10 of these 23 were resolved as HIV-2 non-reactives on retest. For our false positives, again, most of these were multi-spot non-reactive. We had 3 that were indeterminate, and we had 98% in agreement with the negative test on the initial test. As you can, these 4 at the bottom were also HIV, 4 were HIV-2 indeterminate as well, and upon retest of those, 50% of those were resolved as HIV-1 indeterminate, as HIV-2 non-reactive. So it does help by doing the retesting to resolve these prior to reporting of your results. We feel like, yes, you are reporting the HIV-1 negative result in these cases, but with that HIV-2 indeterminate, it probably will cause you to get some phone calls or maybe some redraw bloods. So we've got to prepare ourselves for that and do a little bit more education. Our acute cases, again, the multi-spot was non-reactive, and you can see that all but 3 were also negative, but 3 were indeterminate. So by our standards, these 3 would have been moved to early infections, where we were considering them acutes. And for the early infections, we even saw a more representation where the multi-spot was indeterminate. On the Geenius, 6 of those were HIV-1 positive. Some had HIV-2 indeterminates and as well, but these 6 would have been moved to established infections. So it clearly shows the increased sensitivity, at least in our study, with the Geenius in how we classify whether we're dealing with an acute, an early, or an established infection. These are the HIV-2 results, and I'm about to run out of time, so I'll just go through those. We had 10 samples that were HIV-2 positive. Most were HIV-2 positive with cross-reactivity to HIV-1. So again, they either had that 160 or that 41, but not both HIV-1 reactivity. And 2 of those were clear HIV-2 positives, HIV-1 indeterminates. So in summary, in our study, we repeat testing on our HIV-2 indeterminate results, as recommended by package insert, resulted in 43 to 50% as HIV-2 non-reactive. The global interpretations did not change. I want to make that really clear, that it really affects if you're having to report out both differentiation results, then there is an advantage by doing your repeat as in the package insert. And in our study, the apparent increase in the sensitivity over the multi-spot would reclassify 15% of our acutes and 60% of our early infections as established. And of course, it has the potential to reduce the number of that have to be reflexed to NAT. So I think what we have to do in our situation, our future, is that we're going to be looking at more uninfected individuals to get a truer assessment of the specificity of the Geenius assay. And I think further testing is needed to learn the impact on both the multi-spot and the differentiation test from people that are been on long-term antiretroviral therapy with good viral suppression, and how that really affects these differentiation tests. And we know it affects the NAT, because hopefully you would see good viral suppression. And this is commonly termed proof of positivity in our setting. We're dealing with this pretty much on a monthly basis now. So I think with that I want to acknowledge, thank my Bio-Rad Laboratories colleagues that supported this study and the talk, and back in my shop, Sally Fordon and Megan Lee, who did all the work. So again, here's the documents, and I thank you. And
Okay, I'm pleased to introduce our third speaker now. This is Dr. Michele Owen. And Michele has worked in retrovirus research at the CDC since 1989. She's currently acting as the Senior Advisor for Laboratory Diagnostics in the Division of HIV/AIDS Prevention, where she provides HIV diagnostic subject matter expertise to investigators and members of senior staff within the division, and to many of us out in the community. Prior to the advisor position, and currently she leads the HIV Incidence and Diagnostic Team in the laboratory branch of the Division of HIV/AIDS. The focus of this team is to conduct research to develop, evaluate, and implement new diagnostic testing. In addition to research, she's responsible for the oversight of the domestic HIV CLIA reference laboratory, which provides HIV diagnostic support to state laboratories, epidemiologists, commercial labs, and physicians. And in her spare time, Michele and her husband enjoy touring the country on the motorcycle and teaching others to ride safely. And I think that they've been going around to every baseball stadium also, looking for, can they find any place where it says, Mark Pandori actually played here. Okay? So with that,
Thank you. Thanks, Bio-Rad, for asking me to speak. I think I've done one of these before, so I think this is the second time I've actually got to do this. So I am going to be talking about the algorithm. I am going to talk some history and hopefully where we're going now that we have even more new technology that's out there. I just want to say I do want to welcome everyone to Atlanta too, because I'm also one of the few Atlanta natives you'll actually meet. I was actually born here, so welcome to Atlanta. And we actually, hope that we have a new day coming in HIV testing. So why am I talking about the algorithm? Well, my colleagues actually somewhat talked about it. The tests aren't used individually. They actually are used in an algorithm. And actually, since I am from CDC and I am wearing my public health hat, I do need to put this up here, our continuum of care. So obviously, the first part of dealing with the continuum of care is actually the first step, is diagnosis. And we know that that's important, and we also know that we still haven't diagnosed everyone out there that needs to be diagnosed. So I just want to put this slide up there as reference. We are doing pretty good as far as diagnosing. We're doing less well about getting people into care, keeping them into care, and keeping their viral load suppressed. But the first step is diagnosis. So now I'm going to talk about how the algorithm that you've already heard about, how it actually came about. It did not come about overnight. It wasn't just people sitting in a room saying, hey, this is it. My colleagues up here that are on the stage with me actually both played a huge part in the algorithm. So here's the pathway to change, and there's actually something kind of interesting about this slide too. So obviously the first algorithm came about in 1989 and then was revised in 1992. And I also have a very distinct pleasure of rewriting something that my father-in-law actually helped write originally. So it's a weird situation, but I actually am rewriting some of the stuff that my father-in-law did back in the 80s and 90s. So obviously we started with screening immunoassays for antibody. We did the confirmation with Western blot, and we did that forever, it seems like. And the whole point of that algorithm was to maximize specificity. And at the beginning of the epidemic, that truly made sense because we didn't have any drugs that worked. We didn't want to tell someone that was, give them a false positive result. So the whole idea behind the algorithm was to maximize specificity. So obviously there's been a huge evolution in diagnostic technology. We've improved detection, et cetera. So as technology evolved, unfortunately the algorithm didn't quite keep up with the technology for a very, very long time. And then the other huge aspect of this is we really do have a better understanding of transmission, how and when the virus is transmitted, and also the benefits of actually diagnosing people early. So how did this really happen? Well, it actually started with two other documents, a document that I was involved with the CLSI and one that I was involved with APHL, where we really first started out just proposing a whole host of algorithms. And if you actually go look at the CLSI document or the APHL document, there's like 8 algorithms in those documents. But we were trying to take the technology that was there and figure out what really would work. Well, we kind of needed some data after we proposed all of these algorithms, so that's really what took the time. We came up with these particular algorithms, and then we went about to collect data. We actually had diagnostics meetings in 2010 and 2012 where we are further refining the algorithms and actually presenting data. There's been actually 2 JCV supplements where actually a lot of this data that we actually solicited are published. And so if you're ever interested in a good portion of the data that actually inform the algorithm, it's actually in these 2 supplements. And then we actually did go out and have stakeholder input. We actually asked for comments at all of the diagnostics conferences. We wanted feedback. And then finally, we got around to writing the guidelines. Several years later, once again, we had to go back out and ask for stakeholder comments because we are a public health agency. It had to go out to the Federal Register. We had to get comments. We also sent it out for peer review to various agencies. And then obviously, finally, the guidelines was published in 2014. And it really did start in about 2007, 2008. So it really was a long, long process. Mark showed you a version of this slide before, but I'm going to actually step back a little bit and actually talk about some of the tests. I'm going to show a slightly different picture than Mark showed. I think we really are saying the same thing. We're just going about it in a slightly different way. But this is the classic slide of when things become positive, when you can detect them, et cetera. So just like Mark said, RNA is the first thing that you can detect. The P24 antigen is the next, then IgM, then IgG, and this corresponds to the generation of tests like he talked about. And then down here is what is called the eclipse phase. The eclipse phase in general is thought to be about 10 to 12 days, but if you really look in the literature and you really dig, maybe we don't quite know as much about the eclipse period as we really think. It may be dependent on route of transmission. It may be dependent on the viral inoculum that the person got infected with. So I think the 10 to 12 days is a good estimate, but I think we do need some more information about routes of transmission, et cetera, as far as to truly know what the eclipse phase is. Unfortunately, a lot of that data relies on self-report of when people say that they were at risk. So we know from some other surveys that we do, people aren't always real accurate with self-report. But the point is, there is this defined period that at some point RNA becomes positive, and we can do a lot of characterization after RNA becomes positive. So this slide has been used quite a few times, and it's really looking at the same type of thing Mark did, but we took a slightly different approach. Since we really knew when Western blot became positive, we always opted to work backwards because we had a really defined anchor point that we could define, and then we worked backwards to see when tests become reactive. And my goal is to always to get every FDA-approved test on here. I can tell you there's actually one missing because there actually was another 4th gen that was just recently FDA approved. I will say we actually have some data, but it's just not made it to the slide yet. So it will be coming, I promise. But as you can see, if you look at the Western blot, which was our supplemental test, this is when it became reactive. And then all of these other tests become reactive before the Western blot. And that's one of the things that created the diagnostic dilemma. We're out here screening with these tests, and then the Western blot's not positive, so what do we tell people, et cetera. As you can see, as the generations move along, here's most of our rapid tests here, including the multi-spot and the Geenius, and then the 4th gen rapid test. And then I will say here are our fourth gens, with the exception of the Siemens test that's also been approved. And actually, Mark, in our data, we didn't see a difference between when the bioplex became reactive, at least certainly not a statistically significant difference, but it is certainly just as good as the other fourth gen tests that are out there. And then obviously, like Mark alluded to, the RNA test is in front of the combo test. I'm going to step back and also talk a little bit about why this is important. Mark alluded to it, but I'm actually going to point out a little more data about why acute and early infection detection is important. As he suggested, the viral RNA is very high in the beginning, and we know that that really does lead to increased transmission. There's a tremendous amount of data out there, and like he said, both plasma, semen, there seems to be a real correlation. So we know that that infectiousness is there. This is one of such studies that people estimated that it was 10 to 26 times more infectious in the acute stage. And interestingly, the next one is something that really intrigues me, because actually before I was working in diagnostics, I actually was a pretty basic researcher as far as virology. And this study is quite interesting, and actually Beatrice Hahn is starting to show some of the same type of effects in humans. And that is it's not just the titer, but there might actually be something different about the virus particle. And in this particular case, in an animal model, they actually showed that individual particles were up to 750 times more infectious during the acute phase. So it's not just titer, it might actually be something about the virus. So once again, just supporting the fact that there's greater risk for transmission. There's also, since I work at CDC, I actually work with a lot of behavioral scientists, and at one time I must admit, I never really read that literature. But when I moved into the job I am now, then obviously I became much more in tune with the behavior, et cetera. And actually, there is a lot of behavioral data out there that truly suggests that behavior does change once people know their status, and that people that don't know their status particularly may have more partners. They may have more partners that are actually in the acute phase. So there's not only the virus more infectious, but there's actually more chance and exposure to virus. So what are the benefits? So, well, there's obviously the public health benefit that Mark mentioned. And once again, I go back to behavior. And there's been several studies, once again, people reporting more partners. There's more transmission events in the unaware group compared to the aware group, for example. There was recently a paper that came out that was doing modeling that actually looked at some interesting modeling data, and they actually showed that if MSM did not change their behavior after testing, that there would actually be the epidemic would be worse. So they actually went back and did the modeling. So it's quite interesting. But then also, like I mentioned, there's greater infectiousness, there's higher rates of transmission, et cetera. But then the other great piece to this was the work done by Mike Cohen, et cetera, that basically shows treatment can be prevention. And if you actually treat people and lower their viral load, you prevent transmission. So there is truly both a great public health benefit. But one of the most interesting things, for many years we could say, hey, great, there's this great public health reason for diagnosing people early. But just recently, NIH actually did a controls study where they actually showed there truly is individual benefit. And actually the Data Monitoring and Safety Board actually stopped this clinical trial early because they actually showed the individual benefit of diagnosing people early. So now not can we just speculate that it helps the immune system. There's actually now a trial that truly indicates that detecting is beneficial to the individual, and the website link there is to that study. It's actually quite interesting if you go and look at it. As Mark also and Barry talked about HIV-2 infection, we know it's uncommon in the US. We certainly understand and believe that. But as they suggested, it is misdiagnosed. And they've already mentioned the two studies that I was going to talk about, but there's oftentimes HIV-2 is misclassified. And it oftentimes happens after the person starts to deteriorate immunologically, once they start to get sick. So obviously that's not optimal for those few people that are infected with HIV too. It also causes a huge diagnostic dilemma, particularly before there was an antibody test out there that detected HIV-2 antibodies. So when we set out to do the algorithm, we actually had some goals. So it wasn't, once again, we were just sitting there scribbling on a sheet of paper what we think might work. So we made the conscious decision up front. Did we still want to maximize specificity, or do we now really want to concentrate on sensitivity? And we really made the conscious effort to say sensitivity is really now our goal, because there is treatment. And yes, we might misdiagnose a few people, possibly. It doesn't really seem to be a huge, we've made a huge change in that, but we really wanted to maximize sensitivity, and particularly maximize sensitivity in terms of acute infection. Do the tests, and because of that, we asked the question, do all the tests work in the same at different stages of disease? And hopefully from the slide I've shown you and Mark has shown you, we can say that that's not the case. We did want to think about sample type and volume. I am a laboratorian, so I do understand that that type of thing is important. We greatly wanted to increase turnaround time, if possible. And we didn't want to make the cost substantially more expensive. And, you know, we did know that there might be some implementation challenges to changing an algorithm. So those were our objectives. We wanted to improve acute infection. We wanted to accurately diagnose HIV-2. We wanted to decrease turnaround time. And we didn't want to have a substantial change in the cost. So they've already shown you the algorithm. So here it is. Basically, we wanted to start with the most sensitive test that we could serologically. We wanted to differentiate between HIV-1 and HIV-2 antibodies at the second step. And then in those few cases where the antibody test did not confirm, we would have to go to NAT. Well, this is all great. Yes, as you can see, I like motorcycles. And if any of you have seen Bernie Branson give a very similar talk, he used cell phones and computers as the generations. But I decided to opt for something more fun and talk about the evolution of motorcycles as opposed to the evolution of computers and phones. So obviously, we had the first gen that Mark talked about, the second, the third, and fourth. And basically, these are just my little pictures talking about how the various things are detected. And then, obviously, things are continuing to change. And that's my right side over there. So, you know, we can't stop. The algorithm now is about to become maybe obsolete because of the new technology that's actually there. So, obviously, you've heard about the Geenius. So, we have another differentiation assay. And now we have the Bioplex that Mark also talked about. It is random access, so we can differentiate between 1 and 2 and MP24 at the beginning. And there's also other stuff on the horizon. The other things that are on the horizons are simplified NATs, and that can be hopefully used for diagnostics and monitoring. We certainly at CDC have advocated to the companies that are developing these tests that they seek both claims. There are multiple platforms. The first one isn't really a rapid test per se, but it's actually an adaptation of the current qualitative test, but to an automated platform. And then we have several that are really, truly rapid and could potentially be point of care, whether it's the ALIR-Q, the Cephia Gene Expert, the Samba from Diagnostics for the Real World. Roche has been working on LIAT, and then Theranos, which they're out there, let's just say. And then there's qualitative and quantitative, like I said. And the great thing that I'm really happy to see is some of them actually have made the effort to multiplex HIV-1 and HIV-2. So we might actually have an HIV-2 NAT that can be done quickly, which would make my life much easier because then people won't have to send the samples to my lab. So what is the future of the algorithm? Well, Bioplex is changing it, determined, since it detects antigen and antibody. Once again, now that Bioplex can differentiate 1 and 2 antibodies at the screen, now what do we do in the second step? Obviously, Geenius has some additional interpretations that Multispot did not. So once again, we have to think about those in the algorithm. And then the rapid NATs, like I said, are coming. So there's studies already ongoing, I promise, so stay tuned. But here, man, I've just made this a lot more complicated. So what are we going to do now that we have these new technologies coming? Well, obviously, like I said, now if you start the screen with Bioplex, you have a whole host of things that you can get just out of the screen. So, I mean, I think my recommendation, I don't have data yet for some of this, is, you know, obviously if you get an HIV-2 antibody, you would probably still want to go to Geenius or Multispot to confirm that HIV-2 antibody. If you get P24, then there comes the real question, what do I do? Well, most likely we will probably go to a NAT. But currently right now our offering of NAT isn't particularly optimal, so we can hope that those rapid NATs make it to the market pretty soon. Because if you get the P24 only, obviously you should be screening for the virus, and ideally you would go to NAT. Well, we can go to HIV-1 NAT now. I mean, there is the one test that's FDA approved, or a lab can validate the use of you know, an off-label use of the viral load. So that's pretty straightforward. You would come down here. But then what do you do with those HIV-2 indeterminates that now you are going to get with Geenius? Well, ideally, we would go to an HIV-2 NAT. So we can certainly hope that there is an HIV-2 NAT that's coming soon, or I will have even more samples in my lab, and my people that work in my lab will hate me because we will have more work to do. But as you can see, it's not necessarily a simple question. Some of them, particularly this pathway. And then there are the untypables, which once again, ideally I would reflex to NAT if it's available and easy to do. So there's more to come. There's work to do. I guess it's job security. I guess I can finish out my career at CDC working on the next iteration of the algorithm. Hopefully it won't take 8 years. We're trying to be a little more proactive this time. So conclusions. The new algorithm officially was recommended in June 2014. It's hard to believe it's already been a year. Data do indicate that the lab algorithm can meet the desired objectives, and it does work. Like I said, some of my colleagues up here have actually published a fair amount of those papers. It can identify infection earlier. You can have more accurate diagnosis of HIV-2. And I must admit, I'm really surprised that Barry hasn't found any, because I know they found 2 in Massachusetts already. So you know, I mean, it does work. I'm just kind of shocked, but, you know, hey, maybe no HIV-2 infected people have moved to Florida recently. I don't know. It does decrease turnaround time, as Barry indicated. If you look at our data from CDC, when people refer samples to us, our turnaround time has also decreased since the new algorithm's been implemented. And I would say there's not a huge significant change in cost. As I said, the diagnostic tests do continue to evolve. The tests with greater differentiation capability, the Bioplex, the Geenius, the determine, et cetera, and then the rapid NAS that are on the horizon. So modifications, I will promise, modifications to the lab algorithm will come once the data are collected on new technology. And like I said, we have been a lot more proactive. We have really tried to work with all the companies. I mean, I've had long-standing collaborations with Bio-Rad, but I will say I know there's some other manufacturers in the audience, and I think they will all say that I've been very open to collaborating with them and working with them to evaluate their technology as well. So I certainly hope that at least going forward, like I said, it won't take another 8 years to have some updates since the new technology's there. I used the same slide as Barry because I guess I've also been involved in all 3, and we want everyone to read these documents, so I just put them in a different order as far as I put the algorithm document in the middle and the supporting documents on the side, but hey, you know, it's... and then finally, if you have any questions for me, and there's my email address and phone number.